glutamine, and proline. The carbon atoms of proline and argine also come from alpha-ketoglutarate through glutamate. The proline is synthesized from glutamate. The path to argine is similar in most species except the glutamate amino group is acetylated before the aldehyde is formed. Okay, next is cysteine, glycine, and serine. 3 amino acids are derived from the glycolytic or glyconeogenic intermediate 3-phosphoglycerate. Serine is synthesized from 3-phosphoglycerate in 3 steps. Amino acid histidine come from the glucose 6-phosphate as the precursor can change to ribose 5-phosphate. When the glucose 6-phosphate change to ribose 5-phosphate and not result the histidine, but the ribose 5-phosphate change to the erythrose 4-phosphate, so the result is phenylalanine, tryptophan, and tyrosine. When the histidine, tyrosine, phenylalanine, tryptophan we get, so the precursor still the glucose 6-phosphate, not different. Pyruvate is the amino group acceptor in the synthesis of alanine by a transmination reaction. Pyruvate is also a precursor in the synthesis of the branch chains Asam amino, valine, alanine, and leucine. Pyruvate combine with alpha-ketobutyrate in a series of the 3 reactions leading to the branch chains intermediate alpha-keto-B-methylvalerate. This intermediate is converted to isoleucine in a transmination reaction. So, valine comes from the alpha-keto-isovalerate. Its conversion become valine by valine transmination enzyme. Oxaloacetate is the amino group acceptor in a transmination reaction that produces aspartate. The enzyme that catalyzes the reaction is aspartate transminase. The asparagine is synthesized in most species by an ATP-dependent transfer of the amide nitrogen of glutamine to aspartate in a reaction catalyzed by asparagine synthesis. But some asparagine synthesis can use their ammonia of glutamine as the substrate. So all of the amino acid, their histidine, serine, aspartate, glutamate is entered to the citric acid circle with the part different. In serine, enter to oxaloacetate with the 3-phosphate glycerol, with the precursor is glucose 6-phosphate. And the glutamate enter to the citric acid cycle with the alpha-ketoglutarate.
Okay friends, I want to explain to you about amino acids as metabolic precursors. Before, the question is the primary role of amino acids as the substrate to synthesis protein. But some amino acids are essential precursors in other biosynthesis pathway. So, I want to explain about some biosynthesis reaction use amino acid as metabolic precursors. The first one is the product derived from glutamate, glutamine, and aspartate. The first one, glutamate and glutamate are important players in nitrogen assimilation. The nitrogen assimilation explained the previous question. And the second is the glutamate and aspartate are amino groups donors in many transamination reactions. The transamination reaction explained the previous question. And the third is the glutamate and aspartate are required in the urea cycle. The urea cycle can explain the next question. And then the glutamine and aspartate are also required as precursors in both purine biosynthesis and pyrimidine biosynthesis. The purine biosynthesis and pyrimidine biosynthesis can explain the next chapter. The second is the product derived from serine and glycine. Glycine and succinyl-CoA is the important precursors from the propyrines. The propyrines leading to chlorophyll, heme, and cobalamin. And then the serine converted to glycine is coupled to the tetrahydrofolate to methylene tetrahydrofolate and then become deoxythymidylate and methyl tetrahydrofolate converted to methionine. The third is the synthesis of nitric oxide from arginine. So this is the structure of the arginine. In mammals, found the enzyme is nitric oxide synthase, so can be synthesis the arginine from the citrulline. This is the structure of the citrulline from arginine. And then what is the function of nitric oxide from arginine? The function is the macrophages are stimulated neurotransmittan in brain tissue and neurotransmittan to stimulate erection penis. The fourth is the synthesis of lignin from phenylalanine. So this is the structure of the lignin. You know that the lignin is the main important of the wood in the flowering plants, and then the lignin cannot break down in the animal's digestion, but lignin can be break down in the various fungi. And the fifth is the melanin is made from tyrosine. So this is the structures of the melanin can convert from tyrosine to the LDOPA and the L-dopaquinone convert to melanin. And then melanin is the dark pigment can be found in the bacteria, fungi, and animals. But in the humans, melanin can be found in our skin color and hair color.